Thank you very much. It's very touching to stand here and be welcomed like this. I had not foreseen it when I received the initial invitation, which was, I believe, in May. And I congratulate the organizers for the extraordinary foresight in doing this. So I'm very happy to be here and discover the brewing intellect and the concentration on very esoteric matters that really matter very much in the larger picture. So today I wanted to talk about the subject in a rather general way. I received the award together with Richard Henderson and Jacques Dubochet for a particular development. And so I'm going to talk about this kind of methodology, but I also want to talk about what came out of it. So it's a sort of a gamish of different subjects, which I feel entitled to because I have so much time. Normally in lectures, it's a very nervous exercise watching the clock, and that's one of the privileges that one has now. All right. So deciphering the workings of molecules, the building blocks of life with the electron microscope. So I begin with something that might be surprising. Begin with a slide in commemoration of Dr. Ernst Kinder, who was my first mentor at the University of Munich while I was doing my master thesis. It's called Diplomarbeit there. And he discovered with the electron microscope how butterfly wings get their colors. So I found this extraordinary collage here in the internet. Where else would I find this? So you have here butterfly, and now we go up with magnification. Go up here, up there, color disappears. We see scales. The scales themselves have a pattern. So he discovered that all this here is sort of a magic delusion. It is in fact produced by interference of the light. And all this is patterned in such a way to produce this beautiful appearance. So, and this example suggests how we can make discoveries as we zoom in on the parts of an organism with increasing magnification. So here's an organism, here's an organ, a tissue, a cell, an organelle within the cell, and finally a molecule. Now, the universality of life process on Earth paradoxically becomes apparent right at the lowest level of structure, namely on the molecular. There, all of a sudden, we find ourselves related to yeast, and to the mouse or the pig, and so forth. So, and even plants, bacteria, animals look completely different. And when you go onto these dimensions, all of a sudden, they're very, very similar. The life processes are organized in a very similar way, and in particular, molecular machines are running the same way in these different organisms. And we see the most striking evidence of evolution and the evolutionary theory by comparing molecules from different organisms. And so a closer look at the cells shows that the processes are in fact very similar. The way cells divide, make proteins, store energy, move the needed products around. Here's an example of this animal cell, and you see all kinds of organelles that are, you can think of them as little factories that make things that are needed by the cell. And among these are one of my favorite subjects, the ribosomes. You see them as a sort of a granular appearance on the endoplasmic reticulum. And that earned the endoplasmic reticulum the name rough endoplasmic reticulum, and people didn't even know that it was caused by these granules, and each granule is a ribosome. So what can we say about molecules and life? All life processes are constituted by interactions between molecules in the cell. Molecules have unique, intricate structures defined by atomic composition and arrangement of the atoms. Now, if we want to understand life processes, it follows from this. If we want to understand these processes on the most elementary level, we need to know these structures and how they change as the molecules interact with one another. We want to know everything about these interactions on this highest resolution scale. And medicine has been greatly transformed based on the knowledge of molecular structure accumulated during the past 50 years. And that is in greatest part due to X-ray crystallography. Today, there are over 100,000 structures, atomic structures in the public database, which have accumulated over the time. And medicine has become molecular medicine because of this accumulated knowledge. A very small part so far has come from cryo-electron microscopy, but this is about to change. And as the name implies, X-ray crystallography requires the matter that is investigated to be in crystalline form. So one has to grow crystals. And that's one of the main limitations. So right now we stand at the culmination of a technological development that lasted for decades, but went largely unnoticed in the general public until recently. It involved many innovations of instrumentation, of sample preparation, and computer processing, for which this year's Nobel Prize in Chemistry has been awarded. As a result, many molecules which form the basis of life function in the cell but cannot be crystallized can now be imaged with unprecedented clarity, even at near atomic resolution, in a range of 2 to 3 angstroms. And so their actions, the action of those molecules as molecular machines, can now be analyzed and captured in movies. In this lecture, the challenges and milestones leading to a new era in structural biology and eventually molecular medicine will be illustrated. So these are examples. These are sort of cartoon drawings of molecular machines. We have a machine that makes ATP. ATP is sort of the energy currency in the cell. It's being made all the time. The proteasome, it recycles proteins, it digests them and spews out amino acids. RNA polymerase is required to copy DNA into messenger RNA. A ribosome makes proteins according to genetic instructions. A chaperone folds proteins. Dynein or kinesin transports molecules. Spliceosome edits mRNA. Flagella motor rotates flagella in many bacteria. So X-ray crystallography requires a crystal. The X-ray beam is put on it, and then we get a diffraction pattern. The diffraction pattern is shown on the right. In this case, is I don't know what it is, but it's a complicated molecule. So crystals, crystal means that many copies of the molecule are arranged in regular order. And in X-ray crystallography, we are talking about three-dimensional arrangements of those molecules. And when the crystal is exposed to the X-ray beam, we get diffraction pattern, and then we can determine structure. There's something like a phasing procedure, which is too complicated to go into. The X-ray beam must be high intensity, crystal must be almost perfect. And today, as I said, there are more than 100,000 structures solved. Now come the downsides, and I have listed them in red. The crystal packing means, the packing of molecules in crystal means that an energy minimization is taking place, which means that if we have flexible molecules, then they arrange themselves in such a way that the energy is minimized. That leads to structures that are not necessarily relevant for the functional analysis. Further, many molecules do not form highly ordered crystals, and that's really what EM is solving now. And the sample quantity, that's a very serious problem, because many crystallization efforts consume very large quantities of the substance, and which is not always available. In cryo-electron microscopy, very minute quantities can be sufficient in order to get the structure solved at the atomic resolution. So electron microscopy, it can be used, as you now know, it can be used to solve structures as well. We're dealing with transmission electron microscopy, which means that we have two-dimensional projection images, and they are formed at very high magnification, for instance, in the area of 30,000 to 50,000 times. To reconstruct an object, many different views must be collected. And a sample must be very thin because electrons are very quickly absorbed. And then we also need to be in vacuum because electrons are even absorbed by a very thin layer of air. And now, there is now a downside with using electrons. They cause very strong radiation damage, and so we need to go down with the electron dose. What it means is that we have a very noisy images which are not clear enough to see anything. So, which means that and some kind of a process of averaging is always needed. This is an example of a machine which used to be the most recent one, but now it's sort of an old hat. There's the Polara microscope, but it's done beautiful service to us. The newer generation are the Krios Titan microscopes, and we will have access to a number of them. So this essentially very lowly apparatus, it used to be manual, and now we have sort of fancy version of it. Is a Plunge-Freezer. So that's sort of a central part of the specimen preparation. We have the EM grid is held by tweezers, and the tweezers are held in such a steel rod. And now a small quantity of the solution in which the molecules are suspended is put onto the grid. And then, very importantly, the excess liquid has to be blotted away so that we get the right kind of thickness for electrons. And the thickness is 100 nanometers or 1,000 angstroms. So it's very, very thin. So after the blotting, the steel rod is lowered or plunged so that the grid is plunged into liquid ethane, which is on liquid nitrogen temperature. The reason why it's not plunged into liquid nitrogen itself is that we want to have very, very fast heat transfer. If we plunge it into liquid nitrogen, then air bubbles immediately form at the interface, and they don't conduct heat very well. So that's why we have this, this arrangement of liquid ethane container, which is inside of a vessel of liquid nitrogen. And the purpose of this freeze-plunging is to essentially immobilize the molecules, but at the same time, at these low temperatures, the radiation damage is reduced. This is the result of the fact of the low temperature in a following way. If we cause radiation damage, then the molecule is sort of, you can think of split in some way, but the real damage occurs when these split parts sort of move away from each other. And at very low temperature like this, they simply stay in place. So we have radiation damage, but we don't see it. We don't see much of it. So this is sort of a copper grid, and the entire dimension is 3 millimeters. You can see the grid pattern itself, and then you see this pattern in the background. These are tiny little holes in carbon film, where the carbon film is across, spanned across the grid. And each of these tiny holes here, which are in regular order, is then covered with molecules, which we can image. And I can, at the end, I can show you a very nice illustration where we go from one magnification to the next, and all the way to the level of the molecules. So this is the kind of image that we get. I should say got, because meanwhile the images are of higher contrast because of the development of these very amazing single-electron detection cameras. So, but this is the kind of quality that we saw at the time when this technique was developed. And you can see this fuzzy pattern. It's also a sort of a salt and pepper appearance, which comes from the fact that we have the dose very low. And then you have some stuff that we cannot even identify, and some sort of look reasonable because they have the size range of ribosomes, if we know what the ribosomes look like. But all in all, you can get the idea that staring at a single copy here will not do you any good. You cannot solve a structure from that. So, and also, these sort of have the handicap that they're single molecules. They're sort of swimming around and then they're frozen. And how do you determine structure from that? Well, this was the time when the ideas came out, when I developed these ideas. I have to show you the context in which they were developed. So the context is that obviously everybody realized that multiple electron microscopy views are needed. Now the pioneering work in three-dimensional reconstruction came from Aaron Klug and David de Rozier. They were both at the LMB, at the MRC in Cambridge. And this was actually the first use of three-dimensional reconstruction in any field. Okay, it was first done in electron microscopy. Later on, it spread out for seismic exploration, for finding oil and what have you. But this was really the first one. It was an absolutely pioneering work. But what did they look at? They looked at a structure which had a very high symmetry. It had a helical symmetry, and they looked at the bacteriophage tail. And when you have such a high symmetry, then a single view can give you all the information that you need. So I would say it's sort of cheating, right? You have a problem, but your problem is reduced, but because of the selection of the object that you're looking at. But however, this whole mathematical apparatus of helical reconstruction was completely developed here at the time. But from my point of view, this just didn't deal with the general problem that we face. Then comes my mentor, who was sort of contemporaneous to Aaron Klug. So he was an X-ray crystallographer. Walter Hoppe is his name. An X-ray crystallographer who had turned electron microscopist. So he had a lot of ideas about reciprocal space and how to apply it in electron microscopy. For him, the electron microscope was a diffractometer that happens to collect phases as well. Okay, so it's a very fortuitous kind of instrument because of the struggle in X-ray crystallography to find phases. Here, you get them right away. You get them for free. So his idea to molecular structural research was to collect many different views in a tilt series, in the same way as we do CAT scan, and turn the detector and beam arrangement around the patient. In the same way here, the molecule itself is now tilted in different direction, and each time we collect a projection. And then in the end, we have to reverse this procedure by using a three-dimensional reconstruction algorithm. And the problem, of course, is that in the process, we get a very high accumulation of electron dose. So for 1,000 electrons per angstrom square, you can imagine that in the end, you have nothing to look at. You know, you can reconstruct a molecule, but it's going to be meaningless. So essentially, my whole approach had to deal with this completely wrong approach. So it was developed in contradistinction to what he was doing. Another context is this here. Richard Henderson, Nigel Unwin, Richard Henderson, the name now sounds familiar, and together with Nigel Unwin, made this really stunning progress in reconstruction of a two-dimensional crystal. And this was a crystal of bacteriorhodopsin, which occurs in the purple membrane of certain bacteria. And so the breakthrough really came from two things. One of them is they found a way of embedding this protein or this crystal in glucose, which was essentially sort of a medium which was very similar to water, but obviously in a solid form. So that was one. And the other one, which was even more important, was to go very far down with the dose. They went down with the dose to less than 1 electrons per angstrom square. And with the result that the images were completely blank. You could look at them, there was nothing to see. It's only when you took the Fourier transform that you saw the reflections. And then you used the reflection and also combined them. So they took the phases of these reflections, combined them with the electron diffraction amplitudes. And then, so they got the first reconstruction of the purple membrane protein. At the time, it was 7 angstrom resolution. But at the time, we are talking 1975, that was an absolutely stunning achievement. And everybody wanted to do Unwin and Henderson. There was even a verb for it. Okay, but it soon became apparent that this was a very fortuitous protein. It was very stable, and glucose proved to be very difficult because it really matched the contrast of protein very closely. And so you didn't get the contrast of the protein that you were looking at. So in many instances, this kind of approach didn't work. So this is exactly 1975 when I published this paper, which is sort of a concept paper. You notice one thing: ultramicroscopy was in its volume 1. You might say they were desperate for contributions. But anyway, so this is what I said. The investigation concerns the possibility of extending to non-periodic objects the low exposure averaging techniques recently proposed for non-destructive electron microscopy of periodic biological objects. So, and then the explicit reference to Unwin and Henderson. So the idea is if it works in a crystal case, we are spreading out the exposure over many repeats of a molecule. Then we should be able to do it with single molecules as well. And the only difficulty is, of course, that they are, spread out. They are not in touch with each other. They don't have any crystalline order, which makes it difficult to find the repeats. And even worse, the orientation of each is different. Okay, so this is essentially now the situation. Orientations are different, but that is actually a virtue, because now we get them all. We don't need to tilt. This here, the Unwin and Henderson method required tilting. And of course, you wouldn't tilt one single crystal because you would accumulate the dose, but rather you would just take different parts of a specimen and tilt them in different ways. And then in the end, combine all the information. So in a more formal sense now, I can simply present the idea. The single particle techniques, the concept is this. The structural information is collected from images of single, which means unattached molecules in many copies. And the molecules are free to assume all naturally occurring conformations. The molecules are randomly oriented, so we essentially we don't have a data collection problem. A single snapshot may already give us hundreds of particle views. And if we say we want 10,000, then we simply take as many pictures as are needed in order to get that number. And as we collect more and more snapshots, more orientation will be covered until we have enough for reconstructing the molecule in 3 dimensions. Okay, so now that's very well and good, but what does it mean? What problems do we have to solve? There is an entire work agenda. And so projection images of the molecule need to be aligned to one another. So we have to have a common reference. And we need to have some kind of a concept or measurement of resolution. Measurement of resolution is sort of comes for free in the case of the crystallographic approach, because we see immediately how far out in Fourier space the reflections extend. And we immediately can attach a number to this. But if you have aperiodic molecules, they have no symmetry, they have no internal reference, then it's a lot harder. And then, which is of course the worst problem now, to do a reconstruction, one needs to know the angle of each projection. And in this technique, essentially the angles have to be determined without knowing the structure. If you knew the structure, you wouldn't do the experiment in the first place. So this is how a chicken and egg problem that need to be solved. This is really the hardest part, to determine orientations. Yeah, molecules, as I said, have no symmetries. If they had symmetries, then you would have some kind of an internal reference which you can use for the alignment. And then, of course, there's this nagging question, what if my assumptions are wrong? What if the molecules are not truly identical? Then how we can apply the technique? So there's intrinsic problem of heterogeneity. And then there comes another problem, because if you face a very large sort of a program, and you don't even know what the next step is going to be, because it depends on discoveries that you make along the way. So you cannot write some kind of a master program that is going to solve all the problems. And so, so the idea here is to avoid rewriting a program. At the time when I was a graduate student, I saw other people write computer programs. They were, of course, very limited by the machines that we had available. But nevertheless, a program typically became sort of inflated as the work progressed, and it became larger and larger. It had many bells and whistles and commands and so forth. And it was, of course, not documented, and there was no way of handing it from one student to the next. So all these programs really sort of collapsed in terms of functionality. And so the answer here was a modular image processing program. It had to be modular, because each time you solve the problem, you just had to put it away and say, this is a box now, okay? I don't need to solve this problem anymore. And if I standardize the input and output, then it can shake hands with other modules that come along, okay? So this was one of the first modular programs. I had encountered a modular program while I was during my postdoc period in in the JPL, Jet Propulsion Laboratory, which is part of Caltech, but sort of separated. It's a federal facility. And they were processing images from a Jupiter mission. It was, I believe, the first Jupiter missions. And so they had a very well-developed professional modular program. I think it was called VICAR. And it was very impressive. They had sort of a modularized way of dealing with contrast enhancement and shifts and rotations and what have you. And I learned from that environment how to approach the problems that are faced in electron microscopy in a similar way. So these are just examples. Some of the operations, there were really, in the end, hundreds of operations. Simply a command AC autocorrelation would invoke a subroutine that performed autocorrelation and would take an input image and then produce an output image, which was the autocorrelation matrix, cross-correlation. This is very important for image alignment, Fourier transform, rotate, shift, you name it. Many, many things that needed to be solved so that one can put them together in a script. And so you could develop a program on the higher level just by putting these these commands together. So nowadays, when you look up Wikipedia, then you find all these different things here: Appian, Bsoft, Cyclops, Eman2, Eman, EOS, FreeAlign I added because for some reason it didn't appear here, Imagic, and so forth. Okay, so all these are a modular system, and this one here, which I first published in 1981, is still around and has been widely used. So, in terms of alignment, how do you align images? Well, images are very noisy, and how do you align them? Then I thought about the two-dimensional cross-correlation function and tried it out. That was already during my thesis. And then I found, but to my astonishment, that even the resolution of that instrument was only 3 angstroms. I could align 2 images of a carbon film to a precision of 1 angstrom. And so that gave me, this sort of reinforced the idea, well, now I have I have a tool in order to get molecule images into a register. Resolution, I mentioned before. How do we find out how far the resolution extends? And one can only define it as the resolution of reproducibility, meaning if you form an average of a certain number of images, we could split the images, the image set into 2 subsets and perform the same operation on the 2 subsets and then compare the results. You can compare the results in real space and then you get some kind of subjective measure. But if you go into Fourier space, then you can actually compare the information on successive rings in Fourier space. And you can see how far out you get reproducibility. So that's sort of the idea. And here, this is what got me started thinking in this way. Because if you, thank you, if you take 2 images and put them, align them in the optical diffractometer, if they're perfectly aligned, then you simply see the you see the tone pattern, which is a pattern that is created by the contrast transfer function. But if these two images of the same field, if they're slightly misaligned, if they're shifted against each other, then you see this pattern here, which is a Young's interference pattern. And that Young's interference pattern can only extend up to the point where the information is identical. It cannot extend outside of it. So essentially, this here separates signal and noise. You can see how far out the signal is reproducible. And that gives immediately, this can now be quantified and can be expressed by something like Euclidean distance or phase residual or Fourier ring correlation. All these are measures of reproducibility that rely on the same kind of approach. So then having an approach of alignment and averaging, we have sort of a proof of concept. It was published in 1981. This is an image of HeLa ribosomes. So there's human ribosomes, and this is just a small subunit. And at the time, only negative staining was used. There was no cryo-EM. There was no embedding in ice. So the negative stain was a method of creating contrast for the molecule. And so the molecule that you see is essentially an exclusion image. It's a stain exclusion image. And if you use negative stain, then you get preferred orientations. And so we call them L view and R view, right and left-handed view. And there's no view in between, because they just don't like to stand up in the negative stain. So we picked the L views and formed an average over 81 of these. 81 was a big number at the time. Nowadays, we're talking about tens of thousands, hundreds of thousands. But that was good enough for science to publish it, and because it was a demonstration of what could be done with this. We got a very crisp, relatively high resolution, a reproducible image of the structure that we're looking at. So that was essentially sort of the beginning, because that produced a lot of motivation and a lot of good feedback and ground support and so forth. So now, but I already mentioned there's the problem of heterogeneity. When we talk about two-dimensional analysis, the molecules are presented in different orientations, and then on top of it, they might have different conformations. They might even have different compositions. And how do you sort all this out without imposing subjectivity? So the solution came at a time when a student visited me, Marijn van Heel, visited for a few weeks, who was interested in this whole heterogeneity problem. He brought with him images of hemocyanin. Hemocyanin is a very interesting molecule in worms and in snails and so forth, which substitutes for hemoglobin. So these creatures have hemocyanin instead of hemoglobin, and the metal is copper in this case instead of iron. And you see how it is built in an oligomeric way out of building blocks. And such a complicated molecule then has different appearances when you flip it around, but it also rocks because it has different, it's sort of skewed. And so we have a very difficult sorting problem. We found a way of doing this and produce clusters for the different images. What was behind it was really a pretty amazing encounter. There's a person, his name was Jean-Pierre Bretudier, and he was working at the same place where I was working, but he was a specialist in laboratory medicine who used statistical methods. He used multivariate statistical analysis. And specifically, he used correspondence analysis. And so, as we sort of battled with how to develop programs to cope with heterogeneity, we saw that that he was printing out these images, these pages and pages with clusters, and then we asked him, what are you doing? And then he said, well, these are blood samples, and each blood sample is defined by a series of measurements, such as cholesterol level and so forth, okay? Whatever so laboratory turns out. And now, if you want to compare them with each other, then you do this analysis. And then I told Marine, stop what you're doing, we have the solution. And the solution was simply that the patients or the blood samples of them are images, and the measurements, like cholesterol, are pixels, okay? So all we had to do is to code our data as if they were patients and measurements, and plug it in, and it was a single afternoon when we already got this kind of result, the result that I showed you before. So there was sort of an amazing coincidence, and that was only possible because at the time there was a single computer in this entire institute, and it was in one room, and everybody had to go into this room and was forced to talk to each other because it was so it was so boring in this noisy room. Okay, so this is the nature of scientific discovery. Throughout my entire life, I've been fascinated by these kinds of encounters. You cannot predict them. You simply have to walk around and have your eyes open. You have your eyes open for all these coincidences. So now comes this other invention that Jacques Dubochet has been credited for. Some of the credit needs to go to Robert Glaser, but we are running out of numbers as usual. So in 1976, together with his student Ken Taylor, he already developed frozen hydrated technique of specimen preparation. Now, what he didn't do was he didn't freeze rapidly enough to keep the molecule in this embedded in this really glassy kind of water, or vitreous. We call it vitreous ice. Okay, so without the technique that that Dubochet perfected, we wouldn't have come where we are now. So now, how do we find orientations? And there's essentially a way we have to differentiate between two different phases. One is some kind of initial rough reconstruction, and the other one is some kind of a refinement. So the problem is how to find this initial reconstruction. And what we came up with and I'm talking about Michael Rademacher and myself. Michael Rademacher was one of my first postdocs, and he was very instrumental in the development of this method. There was only existed as a concept that I sort of mentioned in a footnote before, but it was the programs, do this in programs, was very sophisticated. But so the idea which I put on an overhead at the time was that you have molecules when they are untilted, and you look at the molecules that face the grid with the same orientation, and then they differ in azimuthal angle, but they don't give you any new information. But if you tilt this field by a large angle, just a single tilt, then all of a sudden these different azimuths produce different conical angles in the three-dimensional projection geometry. And so that was the idea, because with such a tilting experiment we had everything. We knew the azimuths from correlating the molecules in the 0-degree field, and we knew this other angle because we had just created it by tilting. So we had the theta and phi of a conical system, and that was sufficient to get at least a rough reconstruction. So we had some kind of bootstrap. And these were the first reconstructions. Were actually already produced in 1986, but then in 87 we got a real good EMBO journal publication out of it. So this really solved the problem of how to get three-dimensional reconstruction, how to find the orientations. And then comes an iterative angular refinement. This is really sort of the engine that drives all reconstructions nowadays. Because once you have an initial reconstruction, you can produce a library of projections, of predicted projections, and compare them with the experimental ones. And then, so by this comparison, by cross-correlation, you find a better angle for the experimental ones. From these projections with newly assigned Eulerian angles, you make another reconstruction, which is hopefully better than the one that you started out with. This is now the origin of a new reconstruction for a new production of a library of projections, and so on. Now you go in a circle until you get some kind of a saturation and you don't get any improvement anymore. So altogether, there were essentially milestones. Somehow that yellow path is lost here. That must be resolution of the whole system. So it starts over here and sort of worms its way here. And, you know, I sort of put these different milestones in the methods development right here. One important one is the TDF correction. Contrast transfer function is sort of a malaise of the electron microscope, which everybody has to fight against. So it complicates matters. And so this solution was very, very important. And then maximum likelihood 3D classification, that now deals with the heterogeneity in a serious way for an entire data set. So I just wanted to show you some structural information that came out of this. One of the first structures where we used the cryo-EM for was the calcium release channel. And that has significance because most recently I got into a collaboration with people at Columbia University, where we got too close to atomic resolution. So you appreciate that here, this 45 angstrom resolution at the time, produced great excitement. You saw features that were not seen before and so forth. So that was one of the first applications. And in the ribosome, of course, we got steady progress with it. And this was an absolute breakthrough with data that were collected in a microscope in Heidelberg and by a very bright student. So here we see for the first time the ribosome, these ribosomal subunits with a very intricate topology. There was 25 angstrom resolution. We see inter-subunit bridges. And from these features, from these topological features, we could infer what the likely path of the mRNA would be and where the tRNAs would be sitting in a fully functional ribosome. And this was really inferred from biochemistry, from many other observations that were published before. But we were dead right with this inference. It's true up to now. This is exactly the placement of mRNA, of course, just at that resolution. This was to me, a very satisfactory breakthrough. We collaborated with Günter Blobel, and who had been working on the translocon. And here we had a eukaryotic ribosome with the translocon attached. And this was the first three-dimensional image where you actually see the translocon with the Sec61 complex. Oops, what happened? Okay. And I very quickly have to go through this now to tell you how proteins are made. The ribosome has to translate from the language of the genetic code to the language of amino acids. And the translation is accomplished by the ribosome with the help of the tRNAs. The tRNAs have something that recognizes one language on one side and produces the words of the other language on the other side. Okay? So the recognition of this code here induces a particular sequence of amino acids on the other side. So that's the idea. And if we look at this with increasing resolution, we see that we saw the large subunit here. We see the small subunit with the tRNAs at different sites. The tRNA enters the so-called A site and moves over to a P site, and then exits at the E site. The elongation cycle of translation is we were able to put this together from different complexes that we were visualizing at the time. So that you have to imagine that for each amino acid to be added to the polypeptide chain, we have to go around one cycle. And so the tRNA is incorporated if it's the correct match, and then we need to find a way of moving the entire mRNA with the attached tRNAs over by one codon. And this is a very interesting process because it involves a very large transport of the messenger RNA and the attached tRNAs by one codon. So the question was always in everybody's mind how the ribosome accomplishes this. This is one of the areas that I focused on specifically in my research. So at the time, we produced a little cartoon using Maya in order to show how a protein synthesis works. So we have the small subunit, large subunit, the mRNA. Okay, now comes a tRNA and finds the correct one. It is accompanied by EFTU, the elongation factor TU. Now we have a formation of peptide bond. EFG moves in, that's another elongation factor, which catalyzes this translocation. Now we see another tRNA come in, okay? So this was really, at the time, I think we made the movie in 1999 or so. That was very instrumental in people sort of thinking in terms of the process in 3 dimensions. And it's been widely copied in textbooks. What actually happens is that one single message is read by several ribosomes in a series, so-called polysome. So at any given time, you have different lengths of polypeptide depending on how long the message has already run through the ribosome. So the discovery of the ratchet motion was of particular significance. It was the first time any conformational change was discovered by cryo-EM. So what happens is when elongation factor G binds the one that catalyzes translocation, then it causes this very large inter-subunit motion. The so-called, we call it ratchet-like motion, because it looks like a ratchet. So what happened now was this really event in my life. After the discovery of this and seeing this on the computer, we didn't have these fancy movies so far, but I remember very clearly driving to a Gordon conference. It's one of the places where there's a discussion forum for 3-dimensional electron microscopy. I drove there, and all of a sudden it occurred to me that if eukaryotic ribosomes behaved in the same way, then in fact, every single cell in every single leaf, on every single tree that I was passing by, there were these motions, okay? Tens of thousands, trillions of them, they were moving, okay? It was sort of an incredible experience, sort of mental acrobatics. So this, to get close to this, a student was kind enough to make such a simulation. And the purpose of the ratchet motion is really to move the mRNA by a certain measured amount, and then we have some kind of an unlocking event, and then the small subunit moves back, and the mRNA stays in place. So we have exactly a motion by one codon. So that's what translocation is supposed to achieve. So I mentioned before the methods of three-dimensional classification. This was a very major milestone because it was now possible for a data set to disentangle the orientational and the conformational differences. And this was, for instance, illustrated here. We have a ribosome which actually performs this ratchet-like motion even in the absence of EFG because of Brownian motion. And so we see that we can extract several structures from the same sample and do a separate reconstruction. And this is entirely different from any other methods of structural research. And when you look at X-ray crystallography, you get a single structure. If you're lucky, you have somehow two molecules in the same unit cell, and they might have a different conformation. But here, we really can get an entire inventory of structures from the same sample. And now I'm going to talk about something different, because we have run with this methods development over years and never gotten to atomic resolution. So at the time, maybe 2010 or so, or even at that time, I was resigned to the fact that we'll never get there. We will never be able to compete with X-ray crystallography. And in fact, what I show here is the best structure that we ever got by recording on film. Recording on CCD cameras would even be worse than that. So we were hitting a wall at around, between 5 and 7 angstroms. That could not be broken. And then these wonderful cameras came along. It was a completely independent development of 3 companies. They were powerful. They're able to detect single electrons, and they actually record movies instead of single images. And they allow you to do drift corrections and even follow the progress of radiation damage. So here is an example of what we get from such an analysis. So we get ribosomes actually in different states of the work cycle. We can order them and put them together in sort of a canonical way. Here is something that goes beyond this. We don't just have in vitro an in vitro system, for instance, for translation, but we can now also go and take cell extracts. We purify ribosomes from cell extracts, and if we do it right, then we get the ribosome in all different states. And this here is an example of different states that we obtained for the Plasmodium ribosome. And essentially, that brings us to today. We got this extraordinary resolution of 2.5 angstrom recently, and that was published in PNAS. I'll show you just to get you an impression of the quality of these kinds of maps. So here we have taken the small subunit away. The large subunit has a higher resolution because it behaves more like a monolithic entity. The small subunit is very floppy. You move in. And I think, well, what my student wanted to show was a density modification due to an RNA modification. So at 2.5 angstrom, we can actually go through the entire structure and find RNA modifications. So more structures. More recently, got involved in collaborations with people who are interested in ion channels. This is the AMPA receptor, and one of my students, whom I co-mentor, has been working on this. And these are all examples for structures that approach the 4 and 3 angstrom range. Here's a rotating structure, the AMPA receptor. And then now we revisit the calcium release channel that I told you before. And right here, I got involved, more recently I got involved in the development of a new method of classification, which no longer assumes that you have discrete structures, but the structures can be in a continuum of conformations. And this is just an example of what we get from this. This goes along a continuum of conformations, and this is not produced by morphing. This is an actual pathway that the molecule takes. So in conclusion, single particle cryo-EM has opened up a new horizon in structural biology, and structures of many molecules that could not be determined by X-ray crystallography can now be readily solved. Multiple structures can be solved simultaneously. I call it story and a sample. A new near-atomic resolution has now been obtained for many transmembrane receptors and channels, and opened them up as targets for drug design. And there's a new frontier. I showed you a single movie of this, the ability to map a continuum of states to obtain free energy landscape experimentally. So now I promised you one very, very quick fly through a grid. So this is the grid, and now we go closer. We see these little holes. We focus on one of those. And in the field, and now the molecules move, okay? They're all in different states. And now at any random time point, we now freeze them, okay? Right there. And then when we image them, they are frozen in one particular conformations for each of the molecules, and they attain this peppery appearance from the low dose. And then we need to have the computer locate them individually, put them in a gallery, and then we need to do a reconstruction from them. So that brings me to the end, the very end of my lecture. I did find, I have lots of pictures, images from the history of everything. I found this image here, which shows just a few of my people and other people that were sort of associated with it. And it could be around 2000 or so. There was an event in the Albany area, and I just want to point out this is a very dear friend, Nicolas Boisset, who died very early, and he had been working on hemocyanin, Carmen Manella in my Wadsworth, in the Wadsworth Center was very supportive all the time, but he was not directly involved in the cryo-EM development. Raj Agrawal joined me in around in the 90s as a ribosome biochemist, and with Raj, we got the first very significant results in the development of, in the research on the ribosome. Paul Pensex is one of the powerhouses. He had in background in electrical engineering, and he developed an amazing variety of image processing procedures. Rasmus Schroeder was the host in Heidelberg when we collected data there. Bob Grasucci is still with me, and he is really the EM and lab manager who now runs the entire facility for Columbia University. Martin Kessel, very dear friend, who joined me very early, and he was one of the first true believers in single particle techniques when nobody else believed it. Michael Rademacher, I have to particularly mention because he was behind the development of the of this very powerful algorithms for the random conical method. Jose Maria Carrazzo has been with me for a couple of years, and he is now, he leads one of the leading image processing labs in Madrid. Manjuli was with Raj, and he has also been involved in the cryo-EM. Montserrat was with me at the time, and she was a visiting postdoc. So this is the group. We every year at a particular time when it doesn't rain, if it's not too hot, then we go up onto the roof of the Black building, the William Black building, and take a shot. So we have now years and years of these, and I think in the year before, I'm holding a ribosome, a 3D print of a ribosome in my hand, but I thought this year I'd just leave it on my desk. And so I've given credit to a lot of people of my group over the years. I just really want to add a very big credit to my wife, who is here with us, Carol Sagernow, and who has been incredibly supportive over all these years. And it's truly, I have to say that with that support, I wouldn't be here right now. Thanks.
Hello. Yeah, that was an inspiring account of this amazing and powerful invention, and also the deep biological understanding that it made possible. So Professor Frank will take a few questions. Let me start with a question from the other room, from the people who were not lucky enough to sit here. So can you couple this technique with a slicing procedure to view conformational changes, ultramicrotomy, at the core of a protein, for example? Is it easy to view conformational changes that are internal at the core of the protein, say during ligand binding? Well,
these were 3 different questions packed in one. Well, maybe one at a time. When I hear ultramicrotomy, then you're talking about sectioning So this is almost the opposite of this technique, because I mean you don't have molecules in solution, so the entire method wouldn't work. But then,
is it easy to view conformational changes that are internal or at the core of the protein?
That in the interior of a protein? Yeah, of course, we see everything because we form a three-dimensional image. So we can, all these changes become apparent. It doesn't matter whether they are on the periphery or in the interior of a protein.
When you are freezing the sample, there is a conformation change?
I'm sorry?
When you're freezing the sample, whatever may be ribosomes, that there
what are the what?
Conformational changes, anything will happen during freezing?
In the process of freezing. I wish I knew, because we are really working on the assumption that we have the same conformation as in the ambient temperature that we have the sample in, and the freezing is estimated, or I think this is a very good estimate. It's 100,000 degrees per second. That's the freezing, but it might still not short enough in order to prevent conformational changes in the process. And this is really $1 million question. How to find out what happens.
When you change the solvent, when you change the solvent, whatever solvent you are using, water, ethanol.
I cannot understand you, I'm sorry.
When you change the solvent, whatever making the dispersion, ethanol.
I'm sorry?
We change the solvent.
Whether it will help?
Change the solvent. Well, but then we have to have a theory of the solvent interaction with the molecule. It's really, I mean, it has to be more conclusive. It has to be based on some kind of measurement directly. Yeah, we have to discuss this further.
Okay, thank you, sir.
There's one more question which says, freezing possibly reduces or stops the biomolecular processes. Is this not a disadvantage of the method?
Okay, can you repeat it?
It says freezing possibly reduces or stops biomolecular processes.
Or stops? What are or stops?
Stops.
Or stops, okay.
Biomolecular processes. Is this not a disadvantage of the method?
Well, it's an advantage because you want to have them stopped. I wouldn't know what to do if they were still running.
Sir, good evening, sir. My name is Srikanth. I am not a biology student, but I have a basic question. So, when you go for finding out new structures, you take different sets of images and use different techniques and ultimately reconstruct and finally arrive at a structure. But since you don't have a database of that structure, how do you strongly conclude that this is the right structure that we have arrived at?
That's very simple. If you get to the resolution of 2.5 angstrom, then you don't even have to know the sequence. You can read the structure. You can go along the main chain and actually read the signatures of the amino acids. So when you are not reaching really very high resolution, then you're really dependent on information that is already in the database. Yeah.
Thank you, sir.
Hello, Professor. Here, this. So we was talking about biological samples, right? My question is a little more generic. We use the same cryo-EM method to stabilize non-biological specimens and just get equally crisp and sharp images?
Well, I know that there has been increasing interest in the material science community, and cryo-EM is actually being used there. But I don't know much about it, and I don't know what the nature of the instability is that they try to overcome. But apparently they are excited now. I know there are people at Cornell that go into that direction.
It's being done.
It's being done.
Done.
Yeah.
Thank you.
Cannot be invented anymore. Okay.
Yeah, good evening to one and all. Professor, my question is related to rocket science. We have liquid propellant engines and solid propellant engines in rockets. But we use propellants as the fuel rich, which is a mixture of nitric acid, kerosene, or N2 or any other form of liquid. Can you please just throw me light on what exactly is the meaning of fuel rich as well as oxidizer fuels?
I'm tempted to say I don't know what you're talking about because because it's so foreign to the area that I'm familiar with. But it's been true. It's been true that there's a transformation of Nobel laureates that in the end they think they can talk about anything with great authority, and I'm not going to go this route.
Still, how many years would it take to compare your experience with the structures you have now, the theoretical data we have, experience we have about the structures?
Yes.
Is it possible to clash or compare and make any developments? Like, can we compare what the quantum mechanics or any other theoretical data says about the structures and what your experience experience says about the same? And is the research going on about the clashes and make any developments?
Are you talking about molecular dynamics simulations or what?
No, I don't know anything about. In a general way, I'm asking: is it we have theoretical data now? Right now, we might be having theoretical data on what the atoms may be, on what the molecules may be from the quantum mechanics and all. But we have got the real experience, that what you are saying now, that we know the three-dimensional structures and all. Is it possible to compare on what that theory says about the same structure and what the real 3D structure is saying about it?
If you predict the structure and we have an experimental structure, then of course one can compare them. Because they are sort of presented in the same units, namely atomic structure. So I don't...
Is it...
Yeah.
Okay, I will...
Okay.
I have two more written questions.
Yeah.
One is, what is the theoretical limit of magnification? A theoretical limit for magnification.
There is no theoretical limit, and beyond a certain point, it makes no sense to magnify more, because you want to get a structure out by an averaging procedure. So, if the question were what's the theoretical limit of the resolution, I would understand it, but not magnification. Maybe that was what the person wanted to know.
Okay.
Yeah. So, yeah, good afternoon, good evening. This is like a small step to understand the molecular dynamics of a ribosome or a cellular machine, as you told. So in future, like for materials as well. So if this goes for a structural biology as a small step to understand the molecular dynamic level for medicine, can we implement this for a higher, like more dynamic, complex, mechanisms that go forth? As you showed the molecular level for ribosomes, how the mRNA is synthesized and how the ATP, EGs go on, can we scale it up to a molecular complex dynamics? Yeah.
I don't...
Can you scale up?
Scale up.
Can you scale up to larger molecules?
So now we showed an organelle function.
Well, I would be very careful with this, because the ribosome in itself is already gigantic. Mm-hmm. And the molecular dynamics simulation have only scratched the surface, because they have not been able to do it long enough in order to discover events that occur sporadically. And so I don't want to scale it up right now. I want to have really much more application to the ribosome itself with very powerful machines. Yeah. Yeah.
Okay.
Yeah.
You could continue that later. So we'll take 2 more questions, and then there is high tea where you could try to speak to Professor Frank. You.
Hi, sir. Myself, Suman. Here. One second.
Please pass the mic to her.
No, I'm asking the mic from.
What is the question?
I'll add a question for you.
No, I'll just ask you a simple question.
Yeah.
Just a simple question, one liner.
Hello, sir. Thank you for your nice talk. So you have shown in detail. Am I audible?
Yeah.
Yeah. Sir, here. Sir, here.
Oh, okay.
Yeah. Thank you for your nice talk. So what I wanted to know that you have shown in detail the structure of ribosome. So like that, if we want to do on any other biological molecules which are in even random statistics, so have you done those other than ribosome? Any other biological?
I showed those structures. I showed the channel structures.
Okay.
Yeah.
Last question.
Yeah. Good evening, sir. It's here, right in the front.
All right.
Firstly, with regard to the main motto of the thing is to ensure the entire structure of the ribosome and creating a drug in order to stop the process of translation in majority of the disease. How efficient is it when you actually find a drug to block the entire process over than actually terminating the entire organism as a whole?
I didn't catch the last one.
How efficient is the process of blocking only the aspect of translation over the process of terminating the entire organism in the alter routes rather than killing the organism through other routes like...
He's asking about a
what
drug
other
which will
routes?
only stop translation versus a drug which will do much more.
Versus what? Versus?
A drug which will only stop translation
translation.
versus a drug which will stop the entire process. Or kill the cell or something.
Both are the same. It stops the entire
Both are the same.
process.
And then there was another point with regard to the Brownian moment.
No,
I'm just...
no, I'm
You
sorry.
can ask us.
I'm sorry.
So there's just one brief question. You said it's very brief, so.
Hi, sir. I would like to know that does this technique can be used to know the conformations in the molecules? Very simple question, sir. Like if you have a carbon and a hydrogen bond, and if the hydrogen bonds are rotating around, the single bonds, so can we use this technique to know the conformations? Can we visualize them?
The hydrogen bonds?
Not hydrogen bonds. Like, for example, you take a ethane structure, and ethane has a conformations. So can we visualize it directly? Can you freeze the conformations?
Are you talking about rotamers or things?
Yeah, rotomers. Yeah, exactly.
Yeah. We see exactly the position of the side chains at the resolution, 2.5. There's no problem.
Yeah. Thank you, sir. That was my question.
Okay, could we thank Professor Frank once again for this?